process that uses glucose to produce energy in our body in form of ATP. This organic molecule is essential for every activity in our body, from muscle contractions to brain activity. In this presentation, we will break down the three main stages of the cellular respiration: glycolysis, the Krebs cycle, and the electron transport chain, and analyze how they contribute to produce ATP.
The first metabolic pathway we will present is glycolysis by teams 3 and 5.
First of all, what is glycolysis? Glycolysis is a process that takes place in the cytoplasm of the cell. The goal of it is to produce ADP molecules to allow other reactions to happen in the cellular respiration process. Also, it is important for the breakdown of glucose into simpler molecules, such as pyruvate. The net yield of this process is two pyruvate molecules, two ATP molecules, and two molecules of NADH, which work as electron carriers.
The first step is the phosphorylation of glucose. Glucose is phosphorylated at carbon 6 by the enzyme hexokinase, using ATP as a phosphate donor. Glucose turns into glucose 6 phosphate, and this step prevents glucose from leaving the cell as phosphorylated sugars cannot cross the cell membrane. The second step is the isomerization of G6P. G6P is isomerized to fructose 6 phosphate by the enzyme phosphoglucose isomerase. This step prepares the molecule for subsequent phosphorylation in the pathway.
In this third step of a glycolysis, fructose 6 phosphate gets one phosphate added through an enzyme catalysis and converts itself to fructose 1,6 biphosphate. The enzyme here is phosphofructokinase, and this step here is known as a committed step because it's heavily regulated.
During this step, fructose 1,6-bisphosphate is split into two 3-carbon molecules, which are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by the enzyme aldolase. You can see this process more clearly displayed on the right.
In the fifth steps, we have the isomerization of DHAP to G3P. The hydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate through an isomerization reaction catalyzed by the enzyme triose phosphate isomerase. Throughout first 5 steps, ATP is consumed as it acts as the donor of phosphates in both phosphorylation reactions, increasing the free energy of each intermediate metabolite.
Now, here's an overview of steps 6 through 10. These steps will be talked about in the next slides by members of Team 5.
In the sixth step of glycolysis, the enzyme, which is glyceraldehyde 3-phosphate dehydrogenase, has two main functions. The first one is that it dehydrogenates glyceraldehyde 3-phosphate by transferring one of its hydrogens to NAD+, and this forms NADH. The second function is that it adds a phosphate from the cytosol to the already oxidized glyceraldehyde 3-phosphate, and this forms 1,3-biphosphoglycerate.
During the seventh step of glycolysis, the enzyme phosphoglycerate kinase transfers a high energy phosphate group from 1,3 bisphosphate glycerate to ADP, forming ATP and 3 phosphoglycerate. This is where ATP is produced for the first time in glycolysis, helping to recover the ATP used earlier.
Here, in the eighth step of the glucolysis, it occurs the isomerization of the 3-phosphoglycerate by the coenzyme phosphoglycerate mutase. This coenzyme it needs magnesium to work. Also, is an isomerase, so that means that it did not remove or addition any phosphate groups, only shifts in position within the same molecule.
In step 9, water is removed from the molecule to phosphoglycerate, where it's turned into phosphoenol pyruvate. This reaction is catalyzed by the enzyme enolase.
The enzyme in this reaction is pyruvate kinase, and it transfers a phosphate to the ADP to form pyruvate, which is the final product of glycolysis.
Now, Team 2 and Team 6 are going to present the Krebs cycle. In this first step, pyruvate is converted to acetyl-CoA by losing one carbon as CO2 and generating NADH from NAD+. Then acetyl-CoA enters the Krebs cycle by combining with oxaloacetate to form citrate. This initializes the cycle, and the enzymes that are involved are pyruvate dehydrogenase and citrate synthase.
As a second step, we have the formation of isocitrate, where citrate must be converted into isocitrate. This transformation is catalyzed by an enzyme called aconitase. The process occurs in two steps. First, a water molecule is removed by aconitase from citrate, resulting in the formation of cis-aconitate, an intermediate compound. Then the enzyme reintroduces a water molecule at a different position, converting the cis-aconitate compound into isocitrate. This rearrangement shifts a hydroxyl group and prepares the molecule for the next step of the cycle.
So now on step 3, we have isocitrate to become alpha ketoglutarate. To achieve this, we can use the help of an enzyme called isocitrate dehydrogenase that catalyzes the reaction of oxidation. With this process, a molecule of NAD will turn into NADH. And if you can see, the molecule of alpha ketoglutarate is only a 5 carbon molecule instead of the 6 carbons that isocitrate has. So the sixth carbon was lost in the process in the form of a molecule of CO2 that you can see on the arrow here.
In the fourth step of the Krebs cycle, alpha-ketoglutarase is turned into succinyl-CoA. This process is carried out by the enzyme alpha-ketoglutarase dehydrogenase. During this reaction, a carbon dioxide molecule is released and NAD is reduced to NADH, which will later be used to produce energy. Additionally, coenzyme A is attached to the remaining molecule forming succinyl-CoA, which will continue through the other steps of the cycle.
Then alpha-ketoglutarate undergoes decarboxylation. This reaction is catalyzed by the enzyme alpha-ketoglutarate dehydrogenase. During this process, another NAD plus molecule is reduced to NADH, capturing high energy electrons. The result, succinyl-CoA.
Succinyl-CoA synthetase catalyzes the addition of a free phosphate group to guanosine diphosphate, creating guanosine 3-phosphate. In the process, the CoA group is released from succinyl-CoA. The resulting molecule is succinate.
Two hydrogens are released from succinate when succinate dehydrogenase reduces FADA to FADH2. The output of the reaction is fumarate.
Now in this step, fumarase comes into play. Fumarase catalyzes a hydration reaction, meaning it adds a water molecule to fumarate. This reaction is highly specific and occurs stereoselectively, which means it produces only one specific isomer, in this case malate. More specifically, the water molecule is added in a way that introduces a hydroxyl group to one of the carbons in the fumarate, resulting in malate as the final product. This is a crucial step because malate serves as a substrate for the next reaction.
Well, in the final step of the Krebs cycle, the enzyme malate dehydrogenase catalyzes the oxidation of malate into oxaloacetate. It's in this reaction where the reduction of NAD plus into NADH occurs. And this captures a lot of high energy electrons that will later drive ATP production in the electron transport chain. And this reaction is also reversible, but we see it being pushed forward due to the rapid use of NADH.
Oxidative phosphorylation happens basically in two steps, which is the electron transport chain and the ATP synthase action. So what's the end goal of all this process is to donate all the electrons that the electrons carriers FADH2 and NADH had acquired from previous processes, and to create a proton gradient in the intermembrane space of the mitochondria so that phosphorylation of ADP into ATP can occur. And all this process happens in the inner membrane of the mitochondria. In complex 1, the electron carrier NADH transfers two electrons to the coenzyme and antioxidant ubiquinone, and this results in the oxidation of NADH into NAD+, and the reduction of ubiquinone into QH2. And all this reaction, what does is pumps four hydrogen protons into the intermembrane from the matrix.
Complex 2 is an enzyme that helps FADH2 transfer its electrons to coenzyme Q, which transfers them to complex 3 and 4 to help them pump the electrons, since complex 2 does not pump them into the intermembrane space.
What mainly characterizes what takes place in complex 3 is that the ubiquinol coming from the previous complex um, donates both of its electrons. One of them goes to cytochrome C, which will eventually move into complex 4, while the other electron is transferred via cytochrome B to reduce ubiquinone, and essentially the energy from both of these electrons being transferred is used to pump protons from the matrix into the intramembrane space, and essentially this helps create the proton gradient, which will then be utilized to synthesize ATP.
So the cytochrome C donates an electron to the complex 4, which is able to pump two hydrogen atoms per electron across the membrane. After this, the electron needs to go somewhere, which is where the oxygen is needed. So the oxygen is known as the final electron acceptor because one single molecule of oxygen, so an O2, is able to accept four electrons and four protons in order to produce two molecules of water, which is exactly what keeps the electron transport chain flowing. Now, the proton gradient that was generated, the energy that was generated in this case, is used by ATP synthase, and this allows for the formation of a high energy molecule, ATP. So, ADP plus an inorganic phosphate are able to combine in order to create ATP. And a total of 34 molecules of ATP can be produced in this process for each molecule of glucose for the electron transport chain. ATP synthase is able to drive this reaction thanks to the proton gradient because the protons that pass through it induce a clockwise rotation. It is not a counterclockwise rotation as shown in my left. This is because the arrangement of molecules and their charges make it thermodynamically unfavorable for it to move this way. So the clockwise rotation then induces a conformational change in ATP synthase catalytic site, and it is exactly what gives the energy to ADP and inorganic phosphate so that they can produce ATP.
Now the total ATP recap presented by Team 1.
As stated throughout this video, cellular respiration can be broken down into 4 steps: glycolysis, pyruvate oxidation, Krebs cycle, and oxidative phosphorylation. Glycolysis involves the breakdown of glucose into pyruvate and takes place in the cell's cytoplasm. Its notable products are 2 pyruvate molecules, 2 NADH molecules, and 2 ATP. Pyruvate oxidation turns the pyruvate from the glycolysis step into acetyl-CoA. It produces 0 ATPs, however, it does produce 2 acetyl-CoA molecules, 2 NADH, and 2 CO2 molecules. It takes place in the mitochondrial matrix.
The Krebs cycle consists of the acetyl-CoA oxidizing into CO2, producing 2 ATP molecules, all in the mitochondrial matrix, as well as 6 NADH molecules, 2 FADH2 molecules, and 4 CO2 molecules. On the other hand, for the oxidative phosphorylation process, it consists of using electrons from the NADH and FADH2 to produce around 30 to 32 ATP molecules. This whole process is done in the mitochondrial membrane, and the main products are the ATP molecules.
In conclusion, cellular respiration is a crucial process that transforms biochemical energy from nutrients into ATP, fueling cellular activities. This process occurs in four stages: glycolysis, pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation, each facilitated by specific enzymes that accelerate chemical reactions and ensure efficiency. Enzymes play a key role in regulating these pathways, allowing cells to control energy production and respond to changing conditions. Understanding cellular respiration and the enzymes involved underscores the complexity of energy metabolism and the vital functions that sustain life.